So the final macromolecule type we're going to talk about are the nucleic acids. Now, the nucleic acids constitute the genetic material of living organisms, which is to say they're basically the blueprint for everything that happens in the cell. So the nucleic acids contain the information and the machinery for making proteins. The nucleic acids together as a group, all this genetic information, are also what we refer to as the genome. Now, there are two types of nucleic acid, and both are important. The first is deoxyribonucleic acid, or DNA. I'm sure you've all heard of DNA before. We're going to take a look at exactly what DNA is, what its chemical structure is, and a little bit about how it behaves. We have a second type of nucleic acid called ribonucleic acid, or RNA, which has a number of different jobs, most of which have to do with reading DNA and actually creating proteins from it. So the nucleic acids are going to be found in some pretty specific locations. The nucleic acids are really contained in discrete places within the cell. So the first and most important place that we're going to find nucleic acids is in the nucleus of eukaryotic cells. Now remember that we have two major types of cells, eukaryotes and prokaryotes. And eukaryotes are special because they have a nucleus, which is a membrane that surrounds the genetic information, that surrounds DNA. A prokaryote is a cell that doesn't have that, still has DNA, but it's not protected in the same way. We'll also find nucleic acids in mitochondria, in chloroplasts, and in prokaryotic cells. Now really this is largely focusing on where we'll find DNA because the number of different RNA types mean that RNA will travel to other locations in your cell at certain points. So DNA is going to be in the nucleus, also mitochondria and chloroplasts if we're talking about a cell that has those, and then also in prokaryotic cells, but in a prokaryotic cell, it won't be surrounded by a membrane. When we're thinking about the functional groups that we'll see in our nucleic acids, we're going to see hydroxyl groups, we're going to see phosphate groups, so these will be important. The monomers for nucleic acids are something called nucleotides. So each nucleic acid is going to have its own type of nucleotide. So DNA is going to have deoxyribonucleotides, and for RNA, our ribonucleic acid, we're going to have ribonucleotides. So DNA, remember, is storing genetic information, and the RNA is decoding that information in order to build a protein. And if you want to remind yourself about proteins, click back to our last macromolecule set of slides. So I want to take a closer look at our two types of nucleotide. So here we have an example of a ribonucleotide and a deoxyribonucleotide. I'm going to remove my face for a second so that we can look at the constituent parts. Okay, so all of our nucleotides, whether we're talking about DNA or RNA, are going to have a phosphate group, so in yellow on each of these, a 5-carbon sugar, so that's this ring and this ring, and a base, a nitrogenous base, so another ring or set of double rings that include nitrogen within the ring. So we can see that this doesn't just contain carbon, we also have nitrogen in these rings. But DNA and RNA are different, so our ribonucleotides and deoxyribonucleotides are different. And the way in which they differ primarily is the 5-carbon sugar that forms the basis for our nucleotide. So our sugar is this one or this one. And they look mostly the same, other than the fact that this image helpfully colored them different colors. But the major difference that we'll see highlighted here in blue at the bottom. Okay, so in our ribonucleotides, we have a hydroxyl group here on the bottom we have a hydroxyl group here on the bottom. So if we're looking at this sugar, this 5-carbon sugar, is called ribose. And ribose has a hydroxyl group on each of these 2 bottom carbons. Now, deoxyribose is the sugar we find in DNA. That's where it gets its name, deoxyribonucleic acid, DNA. The difference in deoxyribose comes down to, if you look at this word deoxy, the difference is that deoxyribose actually has 1 less oxygen than ribose. So here in ribose sugar, we had 2 hydroxyl groups. But if we're looking at deoxyribose, we still have a hydroxyl group here, but in the second spot, we have just a hydrogen. So just a hydrogen alone. Okay, so that makes this nucleotide different, and that's how you're going to really recognize the difference between the 2 types of nucleotides. It's subtle, unless someone's helpfully labeled and named the type of sugar, but I have faith that you guys can handle this and you can find this difference if you're looking at nucleotides. So I want to dive in a little deeper on the different types of nucleotides. So what I just showed you on the last slide is the basic format for a nucleotide. But I want to show you a little more about how they link together and how they vary in these nitrogenous bases. So what we're looking at here is a molecule of DNA. So DNA is actually a double helix, so 2 swirling, 2 strands together, and we're looking here at 1 strand of DNA. And we can see our phosphate group, that's one part of our nucleotide, our deoxyribose, that 5-carbon sugar here. And here we have our nitrogenous base, so that other ring, the one that contains nitrogen. And we can see that when we link together our nucleotides, we link together these phosphate groups to the sugar, so that's our 1 nucleotide. And then to link it to the next nucleotide, we take basically the foot of that sugar and link it onto the phosphate group of the next nucleotide. So this creates what we call a sugar phosphate backbone that connects our nucleotides and creates a strand of DNA. Now, projecting out away from that sugar phosphate backbone, we have our nitrogenous bases. Now, I have a couple of different kinds, 4. So we have thymine, T, we have adenine, A, cytosine, C, and guanine, G. So these are going to pair together in specific ways. We're going to pair T and A, and C and G. And when I say pair, DNA is always going to exist actually in 2 strands facing each other. So we'll have a sugar phosphate backbone here, a sugar phosphate backbone here. Nitrogenous bases are going to project towards each other, and they're going to hydrogen bond to each other in the center. So if we have a thymine on this side, we'll have an adenine on this side. They'll hydrogen bond like this. Here we can see how that works. So here we see the connection between adenine and thymine. So you can see a hydrogen bond forming here, a hydrogen bond forming here. Between guanine and cytosine, we can see there are actually 3 hydrogen bonds forming. So this hydrogen to that nitrogen, this hydrogen to that oxygen, this oxygen to that hydrogen. And so these nitrogenous bases pair up really because they have spots for those certain number of hydrogen bonds to form. Either 2 if we're talking about adenine and thymine, 3 if we're talking about guanine and cytosine. And we'll also note that we always have 1 nitrogenous base with 2 rings, and on the other side we're going to have 1 nitrogenous base with 1 ring. So in a certain way, thymine and cytosine are more similar in structure. But when we end up with 2 complementary DNA strands, so those are the 2 opposite strands that make up a full DNA double helix, we'll see that DNA swirls, so it's a spiral, just like our alpha helices in proteins. We'll see that this actually keeps that center part of the DNA the same width, right? Like the fact that we have 2 rings here and 1 ring here on thymine that this adenine pairs to means we have 3 rings total. The same is true for this pair of guanine and cytosine. So we have 1, 2, 3 rings across, so that DNA is able to maintain basically a uniform thickness down the double strand. Okay. I'm going to talk for a second about a little bit more about the backbone and how those links between the nucleotides form. So when we're talking about molecules, sometimes we want to be able to refer specifically to specific carbons within a ring structure or within a linear structure. So we give them numbers. Now, you don't need to know how to predict where the numbers are. Really, we're only going to be talking about these numbers as affects DNA, because DNA is really the only molecule this is going to matter for in this course. So I'm going to tell you right now that in a DNA nucleotide, we number the carbon that attaches to our nitrogenous base, the 1' carbon. So this apostrophe is the prime. That's because carbons over here would also be numbered. So this tells us that we're talking about the carbons in the sugar. Okay, so we have our 1' carbon, we have our 2' carbon, our 3' carbon, our 4' carbon, and our 5' carbon is actually this one sticking out that attaches to the phosphate group. Okay. Now this is going to be important because whenever we talk about DNA, we want to tell ourselves or tell each other which end of the DNA has this phosphate group free and floating and not attached to anything, and which end of the DNA strand ends in a sugar molecule. So we're always going to have an end of DNA that is a 5' end, and the 5' end means that's the end where we have this free phosphate group. So a phosphate group that is only bound into this one nucleotide that it's part of, doesn't have covalent bond to anything else. Our 3' end tells us that this carbon is the one that would bind to the next phosphate group. But if we know we're looking at our 3' carbon, if that's labeled, we know there isn't another phosphate group, that this is the last DNA nucleotide. Okay. So here we can see how that bond forms. The bond is called a phosphodiester bond. So phospho we can remember because of the phosphate groups. Diester, don't worry about it. But that's the specific name for these phosphate sugar bonds in DNA. So we can see that that bond forms here between this hydroxyl group on this 3' carbon and this phosphate group that comes off our 5' carbon. And in total, this gives us a molecule that looks like this. So here we see one strand. Here we see its complementary strand. So it's like matching strand, but all the nitrogenous bases are the opposite. And we can see that our numbering, our ends on each strand are opposite as well. So our free phosphate group, our 5' end, is here at the top on this strand, which means that we end with a sugar at the bottom here, which is our 3' end. And the opposite is going to be true for the complementary strand, so for the opposing strand of DNA. So if we have a 5' end here with a free phosphate group, on the opposite side, we're going to have a 3' end with just this sugar. So basically, these nucleotides are kind of like upside down, right? So you can think of these ones standing up. I always think of nucleotides as like a cheerleader with a pom-pom. Sorry, hard to do on this tiny little video. So if we have these right side up nucleotides here, we're going to have upside down ones on the other side. So this would go 3' to 5'. We can see we have these hydrogen bonds forming nicely between thymine and adenine, and guanine and cytosine. Now, we're not going to look more in detail at RNA right now. We'll talk a little bit more about different types of RNA when we get to genetics. But I do want you at this point to be able to tell a couple of differences between DNA and RNA. So one, of course, is just their names, right? This is a table of similarities and differences. So DNA and RNA. DNA is called deoxyribonucleic acid. RNA is ribonucleic acid. And one important difference is that they have a different type of sugar. So DNA contains the sugar deoxyribose. RNA contains the sugar ribose. And we looked, when we were looking at our nucleotides, that ribose has an extra hydroxyl group, whereas deoxyribose has just a hydrogen in that bottom spot on the 2' carbon. Okay. Another difference that we didn't talk about yet is that one of our nitrogenous bases that we see in DNA, thymine, so you can scroll back a couple slides to take a look at thymine. Thymine is not present in RNA. RNA has a different nitrogenous base, uracil, that works the same as thymine in that it will pair up with adenine. But RNA is never going to contain thymine, and DNA is never going to contain uracil. So it's like its own special version of thymine here in RNA. Both DNA and RNA are going to have adenine, guanine, and cytosine, so that's going to be the same between both of these types of molecules. And our final difference between DNA and RNA, which again we'll take a look at more when we go through genetics, is that DNA is always going to have this double helix form. So it's always going to be in these two strands that swirl around each other. Whereas RNA is going to exist in a single strand. So it's not usually going to be found paired up in a double strand. It really only pairs up when we're making copies of RNA. When it exists on its own, it's just a single strand. And we're going to have many different types of RNA with their own jobs. And we'll get to those in the next unit. So now I think we're all good on all our macromolecule types.